Thank you for joining us today. Let me just introduce myself. My name is Pedro Casimiro, and I'm Life VIDALIA Project Manager, and I'll be one of your hosts today. Just some quick notes. My name is Pedro Casimiro. We are recording this session, and we will use some of these contents following the workshop. I would also like to ask our attendees to keep your microphones and videos disabled. Any questions, please use the chat. At the end of each session, we will have a period for discussion. So any questions you have for the speakers, they will be able to answer at this time. So without further ado, I would like to give the floor to Alonso Miguel, the Regional Secretary for the Environment and Climate Change, who will present a welcoming message. Please go ahead.
Hello, everyone. Good morning. On behalf of the regional government of the Azores, I begin by welcoming all participants in this initiative. It is with a great satisfaction that I open this technical workshop on conservation of endemic flora, organized within the scope of the Live VIDALIA project, with the objective of disseminating the results and knowledge obtained during the conservation actions of the target species to other Micronesian projects, organizations, and entities. As you know, the Azores are recognized for their high levels of environmental quality, the state of preservation of natural resources, and the presence of important biodiversity hotspots, including some of the most sensitive, vulnerable, and rare ecosystems on the planet. These aspects take on a strategic dimension for the development of the Azores. Actually, they represent a brand image of the Azores and an extraordinary heritage left to us by our ancestors, which we must preserve and bequeath to the future generations. It is therefore essential to ensure the definition of a solid strategy for sustainable development in the Azores, which promotes a balance between social and economic development and the protection of our environmental quality. In fact, the formation of a government department fully dedicated to the environment, the Regional Secretariat for Environment and Climate Change, aimed to politically signal the importance and the centrality of environment and conservation of natural resources as a pillar transversal to all public policy.
No plano regional, ou seja, no âmbito regional, temos então aqui a primeira série, onde está publicado o orçamento, aqui uma série
I'll just ask okay. Please go ahead.
Okay, I will go on. Therefore, the government of the Azores attaches great relevance to the LIFE project, considering them truly fundamental to achieve some of the objectives set out in the government program in matter of environment, which is why the Regional Secretariat for Environment and Climate Change started to directly coordinate these projects. The LIFE projects represent vital tools for training of the region in terms of protection and promotion of environmental quality, nature conservation, and preservation of biodiversity. And for this reason, they are, of course, fundamental financial instruments which bring with them highly valuable technical and scientific capacity. These are truly strategic projects for the Azores. The Live VIDALIA project incorporates this strategic importance in terms of conservation of nature and biodiversity, allowing to test actions and methodologies to improve the conservation status of habitats and to increase the population of emblematic endemic species of the Azores, such as Azorina vidalis and Lotus azoricus, on 3 of the 9 islands in the Azores, Faial, Pico, and São Jorge, which can then be replicated on the remaining islands and even in other regions of Micronesia. On the other hand, it will allow testing and implementing methodologies for effective control of invasive species of Micronesia. The Live VIDALIA project also has a determining component of environmental awareness and population consciousness for the promotion of behavioral changes involving different entities such as schools and NGOs, which constitutes an important contribution to the success of nature conservation and biodiversity preservation policies, which are obviously priority objectives for the Azores. So I will end this initial intervention by wishing everyone present a good work, and I'm certain that this workshop will be a fruitful sharing of knowledge in the context of preservation of natural resources, and also an important contribution for sustainable development of the Azores and Micronesia. Thank you all, and have a good day.
Thank you very much, Mr. Secretary, for your nice message. I hope all work will go well. I'm sure about it. So we will go on now with the program. So I will now introduce Sandrine Godefroid. She is a senior scientist at Maïs Botanic Garden in Belgium, where she is developing ex situ conservation programs for endangered species in support of their in situ conservation. She is also involved in research on evolutionary aspects in ex situ collections. She is an active member of international consortia dealing with seed conservation and research supporting ecosystem restoration. Before I'll give the word to Sandrine, let me just remind you to keep your microphones in a disabled position so there are no interruptions, please. Thank you. So Sandrine will present us her presentation on translocations of endangered plant species, facts, problems, and needs. Hello, Sandrine. Please go ahead. Thank you for coming today. It's a big pleasure to have you here with us.
Thank you, Pedro, for your kind words, and thank you for the invitation. I'm very happy to be here with you today, and so I'm gonna try to share my presentation. So let me try to share the right one. Okay, can you see it now?
Yes, perfectly.
Okay, great. So I'm going to talk about translocations of endangered plant species. So by translocations, I mean the reintroduction of species or the reinforcement of existing populations. So let me first introduce the topic. Voilà. So you probably know that the reintroduction of individual plants in the wild may be an essential measure to conserve threatened species. And species translocations have been more and more acknowledged in international treaties and legislation, like, for instance, the Global Strategy for Plant Conservation. And as a consequence, it has become an increasingly used conservation approach worldwide. So here in this small table, I have put here some examples of recent reviews of translocations, some of them having a worldwide scope, others only on one single country, but a big country with an impressive number of translocations in Australia, as well in China or worldwide. So this gives you a few examples of translocations that are currently implemented for well, a high number of plant taxa. But the question is, really are successful? And in order to answer this question of successfulness, I would like to first define what success is. It is actually the ability of a population to survive and to reproduce and to adapt to changing environmental conditions. So there are many different definitions that we can find in the literature. But I found one of them that was, to my opinion, more, let's say, more detailed. And the best definition I found so far is the one of Primack and Drayton from quite some time ago. And they say that a reintroduction can be considered truly successful only when a population is expanding in numbers and area, when individuals are flowering and fruiting, when a second and third generation of plants are appearing on their own, and the population gives every indication that it will persist into future decades. Further success would involve the population dispersing seeds into the surrounding countryside and producing satellite populations. So successfulness is not only a matter of being there, just surviving. It's also a matter of flowering, fruiting, producing seeds, dispersing seeds, and also producing new populations in the vicinity. So in order to answer the question about how successful plant translocations are, I used data from the literature, and especially from these 4 reviews. This one having a worldwide scale, this one also, and 2 other reviews from the United States and from Australia. And these 2 are actually chapters from this great book that I recommend to all of you. If we look at the survival of translocated plants, so the first generation establishment, so survival is by far the most commonly reported assessment of translocation success. And if we look at this graph here, showing the survival percentage within a population against the time after reintroduction, we see that this survival rate decreases over the years. And interestingly, the survival rate that is mentioned in the literature, here in dark green, is much higher than the survival mentioned in surveys, so in unpublished documents. So it means that if you are looking only in the literature data, you have a biased view about what survival can be in translocations. Here in this table, it's basically the same way another way to present the same result. Here we speak about time since reintroduction and whether the population is still extant or extinct at the time where it's monitored. So after 5 years, you see that according to this review, 36% of the populations are extinct. And the result is even more impressive after 10 years monitoring, 60% are extinct. So it means that survival rates are usually quite low and it decreases with time. So let us now look at flowering and recruitment rates.
Sandrine, you're muted. Your microphone is off. Sandrine. Sandrine, your microphone is off.
Okay, so I don't know why it's switched off suddenly. So okay, so let me try to go back to my presentation. Okay, also, can you see yes.
it? Can you put on a full mode the presentation?
Oh yeah, okay. Yeah.
Okay, perfect.
Okay, so the number of flowering individuals decreases over time quite drastically. And here in this table, it confirms this situation in another review, just presenting this slightly differently with the percentage of individuals that achieve maturity here spread into different cases using seeds, juveniles, or adults, but in every cases it's quite low. And the percentage of populations where offspring recruited is also quite low. So these two variables are low and can decline over time. So why is this situation? So why are these translocations not successful? Well, there are plenty of reasons for this. For instance, some attempts are carried out in unprotected areas. So you can see here from this graph, if you translocate a species in unprotected areas, it does have an impact on your survival rate. And especially after 3 years monitoring, the situation is even worse compared to in protected areas. So the number of outplanted individuals is also sometimes too small. So this is another reason for failure. Here you see that on average, people use 157 juveniles or 111 adults in translocation projects. This is not so many actually. And it's confirmed by this graph from the Australian colleagues showing the ventilation between different categories of number of propagules that have been used. And usually they used quite few propagules, less than 100, or in the best case, less than 250. Well, this is lower than the minimum viable population size. So what is this minimum viable population size? Well, this is the smallest possible size at which a population can exist without facing extinction from natural disasters or demographic, environmental, or genetic stochasticity. So this is no magic number, or there is no universal threshold around which we can plan a translocation. But from the literature, we see that we do have fitness problems in species when we have populations being less than 500 individuals. And I'm going to give you 3 examples for that, where the exact figure is known. First example is Primula veris from European Flora. Well, the reproduction of this species is depressed very strongly in populations consisting of less than 200 individuals. And a similar example is Gentiana lutea, again from Europe. With this species, the reproduction is strongly depressed in population of less than 500 individuals. And the third example from the literature is for this species, Silene regia. If you have populations of less than 100 individuals, we observe less than 50% germination. And in population of bigger populations with more than 150 plants, we observe a much better germination of 85%. And so this does have an effect on the surviving rate. As you can see here from the American colleagues, so they made a difference between 50 individuals or more than 50 individuals. And here you can see that the proportion of populations that go extinct with less than 50 individuals is much higher than for bigger populations. And this is the same pattern when using seedlings or plants. And here again on this graph, so it's just another way of presenting the same pattern. You have the survival rate of individuals within population and the number of reintroduced individuals. And you see that the survival rate increases with the number of reintroduced individuals. This is a simulation from the Australian colleagues showing that the number of surviving plants over the years increases if you use more transplants. So the blue line indicates a good outcome with 500 surviving plants, and the red line indicates a bad outcome, 50 individuals only, without or with recruitment. And you see that if you use hundreds of transplants, you get a good outcome in terms of surviving plants. And here again, from the Australian colleagues, you see that the probability of recruitment also increases if you use hundreds of individuals compared to very few individuals. Another reason for failure is that seed source material is sometimes collected in depleted populations, so we have to pay attention to the demographic status of the source population. And you see here that when your source population is decreasing, it does have an impact on your survival rate. And the difference after 3 years monitoring is even much more higher than after 1 year monitoring. So if we use a stable population, things are going much better in terms of survival. And this is due to the loss of genetic variation or the accumulation of mutations or increased inbreeding and inbreeding depression in small populations. In other cases, seed source material is sometimes not suited to the conditions existing at the translocation site. And I'm going to give you an example from the Hawaiian archipelago, where you have this beautiful Asteraceae growing there. It is endemic to 2 islands, and it has been reintroduced there. But you have actually 2 different subspecies, although the geographic distance between these subspecies is not that high. You have about 150 kilometers, but they grow on 2 different islands. And so they evolved into different subspecies. So they develop adaptations in response to different local environments. So it illustrates the role of isolation and distinctive ecological conditions in promoting evolution. So it means that if we cross individuals from these 2 subspecies, it might produce progeny that is less fit than either parental form. And as a consequence, we can get what we call outbreeding depression. So it means that when choosing seed sources, we need to pay attention to geographical distance, which is just the linear distance between source and target sites, but also environmental distance, which is actually much more important in an island context. And this environmental distance is the difference in environmental variables between source and target sites. So another reason for failure is that sometimes in the framework of habitat restoration, site preparation is sometimes overlooked, and also post-planting aftercare. So here on this graph, you see that when there is no site preparation and no aftercare, your survival rate is lower compared to when you do this. Also, many attempts use seeds rather than transplants. And so here in this table, you see that the percentage of unsuccessful attempts is higher when using seeds compared to plants. And here again, on this review, we see the same result with another data set. Survival is much lower when using seeds compared to transplants. And here again, from the US, the same pattern appears. Using seeds give a better result. Also within adults, you can make a difference between seedlings and really adult species. So when you use transplants, you have different categories. You have adult plants, you have seedlings, and this also can have an impact on the probability of survival of your population. Of course, seedling is the most sensitive stage in the life of a plant, so it makes sense, of course. And using adult plants gives much better results. So this is an image of what we did with Arnica montana. We use what I would call sub-adults, so small rosettes of about 10 or 12 centimeter diameter. And so we introduced the species as this stage, so a sub-adult stage. And this is just to give you an idea of how it looks like when using these kind of adult rosettes or sub-adult rosettes. The year after transplantation, we already got some flowering individuals, and after a few years, the area is full of flowering individuals. Another aspect is that species biological traits are frequently little known. According to this review, look at this, breeding system is unknown for a bit more than the cases, also past and present distribution. And look at the genetic diversity. So genetic data are rarely available ahead of a translocation. And it does have an impact on your survival rate again. Look at here, this histogram. If you know the genetic variation of your source population before starting a translocation, you do have a better result in terms of survival. Also, surprisingly, species ecological requirements are frequently little known. And as a consequence, you may end up with a poor selection of your translocation site. So this is a graph that we could produce from a survey that we send out to conservationists. And we ask them, what is, according to you, the supposed reason for failure? And many of them, at least 1/3 of the respondents, said it was the wrong habitat. So knowing this, how can we improve translocation successfulness? Well, first of all, I would say that we need to document the species biology and its ecological requirement. And this includes several aspects like reproductive system, morphology, the need for mycorrhizae, the need for host plant, what is the soil seed bank type, what is the dispersal capacity of your target species, its propagation mode, what are the pollinators, the environmental conditions, but also adverse management methods. Then I would say that we need to document the species status and distribution. And this is an example from my country where we first we look at the distribution of a target species, and it helps to select appropriate source material. But it's not enough, because here every black dot might be a large population or a single individual. So we need to go on the field and to look at all these possible source populations, and to look also at the demography of these populations. Here I put 4 examples of populations of Arnica montana, a European endemic, for which we had quite a nice population in 2007 with sometimes 250 or 300 individuals. And after 6 years, we had a collapse in the number of individuals, most probably because of genetic bottlenecks. So this is very important to know in order to collect seeds in healthy populations, because it might have an impact on the seed quality, as I put here on this graph, again for Arnica montana. The size of the population, and especially the number of flowering individuals, had an effect on the proportion of empty seeds. So the bigger was your population, the less you had empty seeds. So you have really an effect in this case on the seed quality. So we also need to collect seeds from preferably large source population with positive growth rate, as I told you. So these are two examples of populations where we collect seeds from. We need to choose the recipient site very carefully, so with sufficient long-term protection, with a known history, ecologically and climatically similar to the donor site, having habitat conditions necessary for the target species, also without known threat, and for which the causes of extinction of the target species are identified and can be counteracted. Also, the site must have current and future land use compatible with population sustainability. And also we may consider landscape level phenomena like topography or ecosystem dynamics. In the framework of habitat restoration, we may consider site preparation before transplantation, like topsoil removal or fencing, because we do not want to have weeds coming back if the site was previously covered with a very weedy vegetation, very degraded area. So we need to consider site preparation. And it's also preferable to use transplants rather than seeds. So really plants and sub-adults especially, or adult species, adult plants. Of course, after that you have developed a protocol allowing the propagation of this species. You may also consider placing your plants in a spatial pattern that will promote pollination, seed production, and recruitment. So this is an example of a plantation design that we have implemented in the framework of a LIFE project. And as you can see, every plant has received a code. And the first letter refers to the donor site. And so we have planted next to each other individuals coming from different origins. And this is because we want to maximize gene flux between plants of different origins, because we know that the pollinators will go from this individual to that one or this one, and not jumping from here to here. So we want to maximize gene exchange. It's also a good idea to maximize the number of outplanted individuals, as we have seen in the graphs. Post-planting aftercare can be important, and especially in areas where you have a high nitrogen deposit. So here in this case, we planted Campanula glomerata. And so without weeding, we got lots of weeds, poppies, so papaver and things like that. So it was not what we wanted. Another example here with Helichrysum arenarium. So without weeding, we got an invasion of rubus species, lots of undesirable species. And with weeding, at least you see the target species. You may also consider sowing accompanying species in addition to your transplants because of a phenomenon that we call the priority effect. This is actually a phenomenon where early arriving species influence the establishment and growth of later arriving species. So in these cases, we transplanted Campanula glomerata, and the sowing mixture was made of all of these species, which were typical species of the target habitat. We need to monitor population demography for a long period. And I would recommend 10 years, because with 10 years, you have a strong data set and you can model the evolution of your population in the long term. And so very frequently I have seen monitorings stopping after 3 or 4 years. And this is definitely too short, because your population can collapse after that. So you may end up with very rapid conclusions that might be false.
Sandrine, sorry.
Yeah.
To interrupt, it seems like your slides are not going forward. We've stopped the image in number 8. How to maximize the number of outplanted individuals. Sorry.
Okay, so let me try to solve the problem. Can you see it now?
It seems like the presentation is
frozen.
Is frozen. Yeah.
Okay. Okay. So well, I'm coming at the end of the presentation. So I don't have. Well, I had 2 slides, and I'm coming to the conclusion because I think my time is over. So what can we conclude from this? First of all, the translocation of plant species is a widely used technique, but it suffers from a generally low success rate. We have seen that variables that play a major role in the successfulness of translocations are material type, number of founder individuals, protection status of target area, demographic status of source population, and management of outplanting sites. The effectiveness of translocation can, however, be improved by following strict rules and striving to build the necessary knowledge of target species, donor sites, but also recipient sites. And given the high cost of translocations and the ever more rapid species extinction, it is also important to share our respective experience in methods and outcomes across the practitioner community. So this was my last slide, but unfortunately you cannot see it apparently. So I would be very happy to answer your questions. And thank you for listening.
Thank you very much, Sandrine. It was really a nice presentation that gave us, well, an insightful vision on these aspects of the translocation. So I will invite our attendees to put their questions on the chat. So if anyone has questions, please put them now. I will also remind our attendees to turn their cameras off, please, and the sound. So maybe I'll ask a presentation. Sorry, a question. Many times in our project we have to deal with very small populations, lower than 50 individuals, and we don't want to mix genetics from different sources. So is there can we be optimistic about the survival of these populations, because they have been there for we don't know how many years, but they are there and they are surviving anyway. So what should we do?
Well, first of all, we need to make a difference between populations that have been rare for a long period, for centuries, and populations that are rare since very recently, since a few decades. So it's really, it makes a difference in terms of translocation. So if your target populations have been rare for a very long period, it means that they probably are still diversified because they are adapted to just being rare. It's like it is. But if you have a population that has been decreasing over the years and decades, then it probably means that your genetic diversity is not very high, and you might also have inbreeding depression. So if you are working on species that are remaining just rare on your target area, you don't have to worry about that. You can indeed select source populations that are just of about a few tens of individuals. You can collect seeds from this population, and you can try to reinforce the population or to reintroduce new populations. But in most of the cases, you have populations that have been seriously decreasing over the years. And then in this case, well, you need to think about your strategy. The best way is to do genetic analysis ahead of your translocation, but most of us cannot afford that because, of course, it's not very cheap. So we don't have the budget for that. You can try to use other methods, like seed quality is sometimes a proxy for genetic diversity. So you can go on the field and collect some seeds in your source populations, and then once back in the lab, you can examine these seeds, and sometimes you can see signs of depletion in your populations. You can have sometimes for Asteraceae empty seeds, or you can have for other families, you can have seeds that are obviously not healthy, or seed weight is another proxy also. So you can try to look at these things. You can do that very easily, very rapidly. It's also very cheap. And then you can have some information at least about the healthiness of your source population. Then if you have several small populations and none of them reach a threshold of at least 50 individuals, then you can consider also collecting seeds from several populations and bulking them in order to start with a number of collected individuals that is high enough to start with a healthy population. But you need to pay attention and to be sure that these several sources are located in areas that are ecologically similar to the target area you want to restore. So the area where you want to reinforce or reintroduce a population. So this is very important. So as I said, ecological distance is more important than geographical distance. And especially in your case, if you have populations on several islands, I would recommend to be very careful. And before mixing them, just studying them very carefully, just in order to be sure that these are not subspecies with totally different behaviors, ecotypes, things like that. So in the context of an island system, it's very important to have an in-depth study of your possible seed sources, source populations.
Okay. Thank you very much. Ricardo Chaves was asking if you can please repeat the name of the book you suggested.
Okay, so the book was, the exact name was plant reintroduction in a changing climate. So it has been edited by colleagues from the USA, and it has been published in 2012. There is a version available on ResearchGate, as far as I remember. So you can download it. If you Google on the title, you will find it on the web. So plant reintroduction in a changing climate. So this is the exact title of the book.
Okay, we have a few more questions, so maybe we can, I will ask you to be synthetic in your answers. Sorry about that. Okay, so Maria Ferreira is asking, what about species that propagate vegetatively? When you have a few individuals in the source population and genetic diversity is hard to achieve, are there any studies on the success for these cases?
Yeah, that might be a problem indeed. So you have indeed several species that do not produce seeds and they propagate vegetatively. And sometimes you have just a clone because, yeah, this is just one genet. And so it's just one individual. You may have several square meters. It might be just one individual. And so starting from that kind of material, well, is very execution for that. Try to find as many populations as possible and as many patches as possible within a single population, because for many species, as I said, they are just clones. So if you have one patch, it might be that it's just one individual. So if you have several patches separated in a population, so it's wise to collect material from these different patches, and preferably also from different populations. But for this kind of, well, clonal species, well, I don't have the solution. It's just, yeah, up to you to decide if you have time and budget to try to save the species. Yeah, I think if there is no other option, if you don't have any other population existing, you may have just one population, you can use that population. But yeah, the key for long-term sustainability of a population is the genetic variety. And so it might be that your population may collapse after a few years because of genetic bottlenecks. But yeah, it might be worth trying it, but it's not an ideal situation.
Mm-hmm. Okay. Thank you. We have another question by Luis Jordão. He asks, as a beneficiary of a former LIFE project, you would probably be aware how evaluation of proposals is not very fond on promoting, funding preparatory genetic diversity studies as part of preparatory works ahead of translocation. So what's your opinion about this, and how did outcomes from the project you managed have been passed to live management or evaluation teams so that such apprehension can be changed, or at least awareness raised for the relevance of genetic diversity for success?
Yeah, I totally agree. Life funding is a great tool, but well, in our case, we had to convince the evaluators that the monitoring was very necessary. That was the first aspect. So after the first evaluation, the reviewers wanted us to reduce our ambitions in terms of monitoring. And so we had to do that. So the problem is that after a few years, EU funding stopped, and then we had to continue the monitoring with our own money. Also for the genetic analysis. So in our LIFE project, we got 5,000 euro for genetic analysis. So you know that it's just nothing in terms of molecular analysis. You can't do anything with that. Anyway, we had 5,000 euro, and we tried to find funding somewhere else, and we succeeded in finding a higher budget. But indeed, in the framework of LIFE funding, it's very hard to convince these guys that monitoring for a long period is important, and we also need to do genetic analysis. So I don't have the solution right now, also especially because LIFE funding is very competitive, and so they probably will not expand the budget for genetic analysis or monitoring. They told to us that LIFE is basically a restoration program, which it is, but they also like some results, and if they want results, we need to monitor, and we need some money for doing that. But in the framework of a LIFE project, I'm afraid most of us cannot do many actions in terms of long-term trend monitoring and genetic analysis. It's like it is, so I don't have any solution for that.
Okay, thank you. Just one final quick question. We are almost over with this question period. Yash Pal Sharma asks, how much is the role of VA fungi, vesicular arbuscular fungi, important at the source and at the side of reintroduction?
Well, that is very interesting question, because that aspect has been hardly studied so far. So that aspect of mycorrhizal fungi in the framework of restoration projects, I have seen only a couple of studies published very recently. So I don't have an answer to that question, but it is a very interesting topic, because I suspect this is extremely important. So if your target species needs some mycorrhizae, we need to know that. We need to know whether it's a generalist species or whether it needs some particular fungi species. And so we also need to study whether these species are present in the restoration site, because otherwise it's not going to work, especially if the species requires these fungal organism. So this is a topic that has been hardly studied in the framework of restoration project, and I would definitely recommend to try to focus on this, because I really suspect that it's a very extremely important question, and probably part of the failures in translocation programs are due to that mycorrhizal aspect. And so, I think this is something that we definitely need to take into consideration. But again, it needs budget, and in the framework of a LIFE project, I'm afraid it's not gonna be possible. But try to search for partnership with universities, with people having a lab who can do this for you. So it might work in this way, not specifically on your LIFE budget, but try to find some partnership in other contexts, and so to complement the results you can have with your LIFE budget.
Okay, thank you very much, Sandrine. It was a wonderful time being with you. So many interesting aspects on this team, and thank you for the questions raised. So thank you. We keep in touch.
Thank you.
Hope
Thank you very much.
Thank you very much. Okay, so I will ask. So the participants they keep their cameras off, enabled, please. Thank you. So I will now introduce Hanno Schaffer. He is associate professor for plant biodiversity at the Technical University of Munich in Germany. He has published more than 40 scientific papers on the flora and fauna of the Azores, where he lived for several years while working on his diploma thesis and PhD project, which produced the first distribution maps and a field guide for the Azores flora. Well, Hanno has always been a reference, I would say even the reference for everyone who is passionate about the Azores and its ecosystems, and you have always been an inspiration for us, Hanno. So I would like to give you the floor now. Maybe you can. Are you there, Hanno? Hello.
I'm here.
Hello.
Thank you very much, Pedro. Bom dia para todos, and hello. It's a great pleasure. And now let me try to share my presentation. How does it work? Actually, where is my screen? Here it is. Sorry.
Okay, we are seeing it.
You see it?
Yeah.
Okay.
Just put it on a wider format.
Yeah.
Please.
So
okay.
now,
Excellent. Perfect. Yes.
So,
thank you.
Yes. Well, you already heard a very nice presentation from Sandrine, and I think that my talk will be much more theoretical because I don't have a lot of practical experience with conserving and reintroducing endemic or threatened species. But still, I hope that maybe some of my thoughts can be useful for you.
Sure.
So, let's see. So I want to talk about the challenges, and of course it will be mainly about plants, but let's keep in mind that when we talk about endemic plant species, especially trees like the Estorian juniper or the laurus, then it also means that we are talking about ecosystems that are crucial for endemic animals, here like this endemic regulus or the endemic buteo, and small plants like this endemic bryophyte here, echinodium. So there's a lot of other species connected to those endemic plant species, and we are going to lose them if we lose those endemic plant species. So it's not just about the Azores juniper or the Azores forget-me-not. There is a whole group of species connected to them. Okay, so I would say there are 5 key challenges for the conservation of the endemic Estorian flora, and many of these challenges apply to floras worldwide, especially on islands. Number 1, habitat destruction. And you will see that, I mean, of course, this has been historically a huge problem in basically all island regions. And of course, also in the Azores, it started with the first settlers. But I'm talking about the ongoing habitat destruction. And I think this is the key challenge that we have to overcome, because as we heard from Sandrine, restoration, reintroducing species is really, really hard and quite often not successful. So it's much better to protect the existing natural habitats than to try to restore something. Then the second, and especially in places like the Azores, very important challenge is exotic invaders. I mean, when Pedro invited me, he said, okay, can you please address a little bit the aspect of these invaders that are overgrowing our restoration sites and what can be done? And this is indeed a big problem in the Azores and in other island regions, that due to human activities, there are now so many very competitive plant species in the islands that are just much faster in growing, that have a very strong propagule pressure, so they produce a lot of seeds, and that can just outcompete the native and especially the endemic species that have never been in competition with these species before. Then the third point, and this is a bit of self-criticism, of course. I mean, I have been working on the Azores flora for more than 20 years now, but there are still so many open questions. And as also mentioned, for many of the threatened endangered plant species, we have actually very little data, biological data, which means things like pollinators, but even things like the number of chromosomes, which is quite important when it comes to mixing seeds or plants from different populations, are not known. So we need a lot more research here. And then a bit in consequence of all this lack of data that we have is this problem of inbreeding depression and genetic swamping. Also things that Sandrine mentioned in small populations, populations that have maybe less than 100 individuals, inbreeding depression can lead to a down cycle so that they go extinct within a decade or so, even if it is a very nice nature reserve. And the last point that is like the Damocles sword hanging over all of us is climate change and its effects, its impact. There are some studies that seem to suggest that it will not affect the Azores so much, but I have some doubts about this. And I will go through all these 5 challenges now in a bit more detail. And of course, yes, you cannot see these challenges as independent, isolated problems. They all interact and they all act at the same time on the endemic species. So yeah, it's a challenging situation. Okay, habitat destruction. Well, on paper, the situation in the Azores is very nice because we all see there's a lot of protected areas. There are these UNESCO biosphere reserves, protected species. So I would say in terms of the legal situation, it's really nice in the Azores and much better than in many other island regions of the world. But we all know when you walk around in the Azores, especially when you have been there for many years, you see the disappearance of natural habitats, the disappearance of individual hedgerows, forest patches. So there is still a lot of destruction going on. And even in the protected areas, we have the problem of overgrazing. And keep in mind that none of the native and endemic plant species has had a chance to adapt to grazers. Yeah, there is no native grazing mammal in the Azores, so none of the native plant species is adapted to this kind of grazing. And this seems to be the reason why rabbits and goats and cows, they really like to feed on those endemics. They seem to have a better taste. They are not as bitter as some of the invaders. This is really a problem. And then even on the roadside slopes, where some of the rarest endemics find their small refugium, like Veronica dabnei, they are in risk of being cut by just the people who clear or clean the slopes. So some pictures. This is the best protected area in Flores, the Zona Central. And as all of you know, it's perfectly legal and fine to have cattle there. Does it make sense from an economic point of view? No, not at all. I mean, my father is a farmer. I can tell you that these cows that you have there and the few goats, I mean, that is nothing in terms of money, but it is a massive threat to the endemic species that sit on these few rocks here in the peat box. So these guys here, they go and eat selectively the few Leontodon and Tolpis plants there. And it's just sad because this can wipe out small local populations, and it's, yeah, for nothing, just for a bit of meat. Yeah, Pico cloud forest. Yeah, clouds you can see here, but it's not the clouds that are supposed to be there. Farmers, again, transforming juniper forest into pasture land. Juniper is protected, and this area is at least partly protected. But when the juniper is dead, then it seems to be fine to burn it, even though this ground here is not at all suitable for cows. I mean, you can try to bulldoze it, but it doesn't make sense. This is from another site in Pico. I could show you similar pictures from Flores. I mean, the farmers don't cut these junipers, but they put so many cows there who like to scratch their backs on the trees that after a few years, the junipers are dead. And I have seen this in the past decade in so many places where these wonderful old junipers just die and are then removed, and then this is bulldozed. And of course, I can totally understand this from the point of the farmer, but we have to really make up our mind if this is what we want. I mean, the milk production goes up in the Azores and the meat production, and all this, or a lot of this, is paid with subsidies from Europe, from Brussels. But this is the same pot of money or the same place where the money for the LIFE project comes from, who then try to restore natural habitats. So I think this is a political decision that, of course, cannot be made in the Azores, but something that should be made in Brussels, where people should really make up their mind and think if it, or decide if it wouldn't make more sense to support landholders and farmers in the Azores to restore and protect natural habitats instead of putting cows in places where the cows are not happy and where the milk production just doesn't make sense. Okay, so that is agriculture, but then there are also other problems. This is habitat destruction on Flores. The locals all recognize this. This is the former massive waste dump site near Ponte Delgada. It was covered up again due to European legislation, who forced, of course, this cover up of this waste dump. But what people didn't have on their screen is that they destroyed with this the largest population of the Azores angelica in Flores. So there were several hundred of these angelica plants in this waste dump site because they like very nitrogen-rich soils. And now all these all this waste dump population is gone. There are still about 300 individuals left outside the waste dump site, but this is really sad and could have been avoided, I think. Okay, so this to the first point, habitat destruction. Now let's come to the invaders. Here I have a small graph showing the number of recorded exotics based on literature data, of course, here, and then in the recent past also from my own surveys. And what you can see here, without going too much into detail, there's a lot of exotic plants coming in. We still have something like 2 or 3 new invaders per year. I mean, not all of them are invaders. Some of them just sit around and do nothing, but many of them are very successful and then just outcompete native plant species. And one example here, Gigante Gunnera tinctoria, came from the Furnas Park probably and was first reported outside the parks in 1966. And now most of you know these large areas in San Miguel where, to be honest, it's probably no longer possible to get rid of it. Yeah. Similar hortensias. I mean, that used to be the symbol of the Azores, of the tourism ministry. But it was first reported less than 100 years ago. And then people started planting it as hedges. It worked well to keep the cattle in the pastures. And now this is a view from Flores in, I think, July. So they are in full bloom. And you can see that there is hortensias everywhere here in these slopes. They even grow up into the rocks where the last endemics are. Here you can see some yellow, maybe Hypericum. And so this, again, is an invasive that is totally out of control, and we cannot remove this anymore. So there are many of these invaders we just have to live with and just have to see how we can get along with them. So here, distribution maps from Flores, from Fayal. In Santa Maria, there's not so much because it's too dry here in the lowlands. But this is definitely a species like Pittosporum or Hedicium where it's too late to try to eradicate them. So we have to live with those invaders. And now the question is, what do we do in such cases as we see it here in the coast on Pico, where Azorina, obviously one of the key target species of conservation, is threatened by invaders. Here Capoprotus with these red arrows, and here Stenotaphrus, a grass species from Africa both. And well, what can we do? The only option is to do small-scale gardening, I would say. So go there and remove those invaders without or trying to cause minimum of disturbance. Because when you do it in a larger scale, as you can see here in Santa Maria at Pico Alto, then you leave a lot of open space. So this is here the rhizomes of Hedicium that have been cut off to remove this invader and to make space for the native bushes and tree species here. But what you can see here is that there's now a lot of open ground. There's nothing. And since the invaders have a much stronger propagule pressure, so much more seeds around, they will colonize this much better than native species. So this is now a real management challenge, I think, to keep this clean. And the only way to turn this into something that is dominated by native species is to put now many seedlings or even larger native trees already, so that the open ground is covered. Yeah, so there's this big problem that we have the invaders very dominant, very present in all ecosystems already. We cannot get rid of them anymore. So when we do some small scale actions, then there's always this pressure from the invaders from the surrounding areas, and it's very hard to withstand this pressure. And the only way is to have as little open ground as possible for them to colonize. Okay, the third point, this is something that also Sandrine mentioned, that we need good taxonomic and biological data. And you all know that in the past couple of years or 20 years, many new endemic species or more broadly speaking taxa have been discovered in the Azores, mainly because everybody uses now genomic methods. And so this is how you can see that there are several endemic bird species, several endemic insects, and also many more plant species endemics than we thought. But from just this phylogenetic data, we cannot really conclude a lot about their biology. So we still need a lot more work here. And just a few examples. This is going down a cliff on Corvu, and you see here those yellow spots. This is Ranunculus cortusifolius. And in theory, this is a species that occurs from the Azores south to Madeira and then throughout the Canary Islands. But there was this study in 2015 where the data clearly showed that there's lots of different genetic entities, and that the ranunculus in the Azores is something very much different from what we have in Madeira and in the Canary Islands. Or another problematic case, Tolpis. At the moment, the official accepted number is 2 native species. One, Tolpis succulenta at the coast. Here, this is Euphorbia azorica in between, which is shared with Madeira, so it's not even endemic. And then one species, Tolpis azorica, in the high altitudes. And Monica Mura from the University of the Azores and Lourdes Borges, and also my group and Dan Crawford from Kansas did a lot of research on Tolpis. And for example, here my student Melanie Schmidt, she studied the pollination biology of these high altitude Tolpis, which are, by the way, highly threatened by goats here on Corvu, in the Calderão, in the protected area, where in winter you have hundreds of cows. And without going too much into details, what we find is that there is multiple independent evolution of highland strains, of highland species in Tolpis. There is one that is only on San Miguel, one that is only in the central group, Pico Terceira, Fayal, and one that is restricted to Flores and Corvu. And then we have here the lowland, Tolpis succulenta. And down here, this is a completely different thing. This is the real succulenta from Madeira. And in the coastal Tolpis succulenta, which is not succulenta, but something endemic to the Azores, we also find, or I should say Dan Crawford found, that there's also differences. For example, he did breeding system studies, and he found that the Tolpis on Santa Maria is self-incompatible, which means it needs to attract pollinators to bring in pollen from other individuals. And this is why the flowers are so large and attractive. Whereas the one on Graciosa, you see, it's much less attractive. It is self-compatible, so it does self-pollination and produces its own seeds, and the two cannot be crossed. So these are also distinct species, which means instead of 2, and one of them not even endemic species, we have at least 3 different coastal species of Tolpis and 3 different or even 4 different maybe high altitude species. And so this means they should not be mixed, they should be kept separate, and each of them is a very precious entity. And you know, these other famous cases that in this example here, something that was thought to be endemic, Marsilia azorica, turned out to be an invasive from Australia, or the other way around, Cadamine hirsuta, which is thought to be endemic, thought to be invasive, turns out to include a clade that seems to be endemic. So it seems to be a mix of an endemic Cardamine clade and something that was recently introduced. Yeah, and with this, I'm almost through. I just want to mention quickly that, of course, these small populations that are often under this minimum viable population size that Sandrine mentioned, they can go extinct just through inbreeding. You see here this Myosotis azorica, where we have in the wild maybe just 50 individuals. I know that Pedro is breeding it and translocating it back into the wild, but it will be difficult because there is no real genetic diversity left. And genetic swamping just means that in the case of this Cadamine or in similar cases, you can then have and the same will probably happen to many of the plant species and also other species that depend on the crater lakes, because we see those lakes drying out more and more often, especially on Pico. And with this, I'm at the end. So my take-home message is, above all, we need to protect what we still have in terms of natural habitats. And for this, I think we really need a change of politics. So it doesn't make sense that European money is used for increasing the milk production and at the same time for restoration of places that have been turned into pastures. This is totally contradictory. And my opinion is that we should better use the money for protection and give the farmers then a livelihood as nature protectors and not force them to produce more and more meat and milk. We need to control invasive species, but local, and try to minimize profit from this. And of course, we need to do ex situ conservation and seed banking as a buffer, as an insurance, in case there's a landslide or a hurricane that just wipes out a population. We have seen this with some of the Tolpis populations already. And we need more research on pollination. Muito obrigado. Thank you very much.
Thank you, Hanno, for such an interesting presentation. It gives us a lot to think about, and many questions raised here for the deciders and well, the more operational teams to come to realize and to apply it on the field. Thank you very much. I'm sure there will be questions. And let me just add that Madalena, where you took those pictures of Azorina, I think it's one of our intervention areas. So we are already trying to deal with it, although they suffered a lot from the hurricane Lorenzo. But anyway, we are intervening in that area. It's hard sometimes to take off invaders like this Stenotaphorum without disturbing much, because they cover the soil with such so much that we had to use machines to take it out. But on the other hand, we used a lot of plants to recover the area. So let's hope we have good results on that.
I think Estorina is a really problematic species because it also seems to like some disturbance. So you often find it in places that are not the nicest ones, where some sewage water is coming in or so. This is where Estorina likes it. And this is, of course, where most of the invasive species also grow very well. And yeah, I see your point. This is really a challenge.
Okay, so we have a question of Joana Betancourt. She asks, how can we preserve the diversity of tallfish species and propagate them on an island where you have different species?
Yeah, I mean, this is really a challenge. And I think at the time being, the safest option would be to keep them on their own island and not to bring everything to Fayal, to the garden, and have them all side by side, because some of them are self-pollinating. Then it's kind of safe, but the others are not. And so you can easily end up with mixing just by some bees or some flies entering your greenhouse or your plots. And this is then, of course, complicated. But yeah, I mean, on the other hand, there is not really facilities on each of the islands. And especially on the smaller ones, like in Corvo or in Flores, it's maybe also, there's maybe not enough manpower then to look after the plants. Yeah, so it's a challenge, but it's definitely something to have in mind, that you can easily have cross-pollination probably between some of them. And even though they are probably not very well compatible, you could end up with some hybrids of reduced fertility, which is not what you want to use for population reinforcement.
Okay. We have another question from Claudia Melo. She says, thank you so much for sharing your experience and ideas. I use your book on the Flora of the Azores as my best field guide. I think we all do that.
Do you have a new edition coming?
Oh, great. Great. That's fantastic. Very well. Do you have any good examples of nature protection that prevailed over the agriculture economy? That's a huge challenge, she says.
Yeah, I mean, to be honest, we see the same problems here in Germany, in my own village. So it's everywhere the same problem, that European money subsidies are given for production on large pieces of land, large numbers of animals. And this is in contrast to a sustainable management and in contrast to nature conservation. So are there any good examples? I mean, of course, you can find ways to bring things together, and there are many places in the Azores where I would say, okay, this is a well-preserved spot, or here money is well invested, like these life projects. I mean, this Estorina life project is, of course, very well invested money, and the Priolo life project, I think, is also very well invested money. But in terms of the agriculture subsidies, I think this is something that goes wrong in the entire European Union. And I think the agricultural ministers, they just met last week or so to try to change it. And I think it was the German minister who stopped it because they just don't want to change it. Yeah, it's really sad, and it's something we all have to work on, try to convince policy makers, not necessarily in the Azores. I know that it's not the Dr. Miguel, the secretary in the Azores, who can change this, but it's something that in the next decade or so has to change, because otherwise we ruin our natural habitats everywhere in the European Union, not only in the Azores.
There is one question that goes on that, exactly on that. So it seems like it's irrelevant for the Azorian biodiversity. It's dependent not on local decision making, but on Brussels policy for agriculture. How do you see now the whole greening announced by the Green Deal to change such situation? For example, it's German population to support payment for ecosystem services as those of existing biodiversity.
Yeah, I think that's a step in the right direction, but it's not enough. Yeah, I think we need to get away from this policy focused on producing more and more. I mean, whenever I talk to a farmer in the Azores, it's about the price of milk and how much milk they have to produce and how they can increase the production of milk. And then when I tell them, yeah, it would be nice to keep the slope here intact and not to cut down the Pau Branco, and then they say, yeah, but I need to increase the size of my pasture because I need to produce more milk. So yes, there are some small steps are being done in the right direction, but I still think it's not enough, and it's nothing that can directly help the farmers in the Azores.
Mm-hmm, mm-hmm. Another question is if you are aware of any other species or subspecies of Myosotis in any of our islands of the Azores.
Mm-hmm. So for Myosotis azorica, I have seen it in the wild now only on Corvo. I know some plants on Flores, but I think all of them have been planted by you, Pedro, and colleagues. So I think on Flores there is probably no more Myosotis azorica in the wild, and if, then it's the same than the one that is on Corvo. But for Myosotis maritima, it's different. So it's present, I think, on all islands, and it is kind of variable. So you find white-flowered individuals, pink-flowered individuals. There has never been a genetic study on them. So it's possible that there are differences, but I don't know. In addition to these two endemic species, there's a lot of introduced Myosotis species. Yeah, but for them, I think it's the same all over the archipelago because they arrived only recently.
It is a very good year, this one, for Myosotis maritima. They are growing everywhere.
Okay. Yeah. Yeah, so someone should start a population genetic study and analyze it across the archipelago. It would be interesting. Yeah.
Another question from Paulo Araújo. He says many precious natural areas in Flores Islands are drowning in Hedychium, for instance, Rocha dos Bordões. And at the same time, money is spent in roadside vegetation management without the least regard for conservation. Do you have any thoughts on this?
Well, I see the same going on in Flores. I mean, I was there first in 1999, and many areas were still cut free from Hedychium in those days because people still used many of the places. And now with all the abandoned pastures growing with Hedychium, of course, Hedychium also keeps spreading into the more natural areas. I know that in the Rocha dos Bordões, the montañeros come from time to time and clean it. But to be honest, I think that is a waste of resources because you will lose. I mean, there's so much propagule pressure, so much seeds of Hedychium transported by the birds all over the island that you can, of course, clean a patch, but after 5 years, you will have the Hedychium again there or even earlier. I think in terms of Hedychium, it would make much more sense to invest a bit more on Corvo, because on Corvo, it is still limited to very few places. And I know that there have been some attempts to eradicate it in the past, but I think it would be possible to keep Corvo free from Hedychium if you send some people from time to time. On Flores, I think it's completely lost. There's no way. And I know from the Priolo people in Nordeste that even with chemicals, I mean, you can kill it in a spot, but it will come back. And we don't want to throw herbicides, huge amounts of herbicides in those areas, and all manual removal. I mean, of course, it works for a short period of time, but then it's back. I wouldn't invest any manpower and any money in cutting Hedychium unless you see in a very focused place that there is, for example, Myosotis azorica growing, and then you clean up a few square meters. But otherwise, I think it's just lost. It's like with hortensias. Of course, you can go and cut them, but it's pointless.
Mm. Okay, one last question. We are a bit ahead of our time. Anyway, has the data on different tallfish species been published already? And where can I find this information, says Sol Eber?
Yeah, so we have published a couple of papers already on tallfish. And as far as I know, Monica Mura and Lourdes Borges from University of the Azores in Ponte Delgada are preparing a treatment for where all these different species will be formally described. You know, it's always collaborations and different partners, and of course, you then have to sometimes wait a bit for others to do their morphological work properly, because they want to really characterize them properly with morphological data. So I think in 1 or 2 years, it will all be formally published. Yeah.
Okay. Okay, Hanno, it's been a pleasure to hear you and to see your presentation. Thank you very much again for this time. So we'll have a small break now. It was supposed to be 15 minutes, but I will give 10 minutes so we can keep up with our schedule. So we'll be back in 10 minutes at 10:50. So let's go for a coffee. Thank you very much.
Yes, of course you can go and cut them, but it's pointless.
Mm. Okay, one last question. We are a bit ahead of our time. Anyway, has the data on we are a bit ahead of our time. Anyway, has the data on Hello again. I hope you're all back from our very short break. So we will restart our session. I hope you can all see and hear me. We will now have a Macaronesian approach of in our workshop. And I would like now to give the floor to Ruli Cauhapi Castells. He is the director of Jardim Botânico Vieira e Clavijo, unidade associada ao CSIC, a research division of the Cabildo of Gran Canarias in the Canary Islands, Spain, and which is devoted to the conservation of the Canarian and Macaronesian terrestrial floras through the knowledge furnished by multidisciplinary science. He is also head of the Department of Molecular Biodiversity and DNA Bank and co-chair of the IUCN Macaronesian Island Plant Specialist Group. He has a strong background on population conservation genetics and molecular phylogeny. So, Ruli, please take the floor. Are you there, Ruli? Hello, Ruli. I saw you on the screen just now, but maybe your camera and microphones are still off. Okay, he is just saying in the chat that apparently the microphone is disconnected. So let's give him a couple of minutes so he can arrange this technical program. It's okay, Ruli. Take your time. While we are waiting, just let me inform you that I'll please ask everyone to keep their microphones and cameras disabled. On the afternoon session, we will have a site on how Life Vidalia works are going on field and also ex situ. Ruli is just coming in the meeting again, so okay.
Hi, Pedro.
Hello.
Can you listen to me now?
Okay. We can hear you and see you.
Okay. Glad to hear that. Apparently, I had some problem with the connection here in the Canaries. Sometimes it happens, but let's hope it won't happen again during my lecture.
Okay, thank you for being here with us. It's a pleasure to see you and to have your thoughts on this issue on the conservation of the flora in the Canary Islands. Examples from the genus Lotus. So please take the floor, Ruli.
Okay, the pleasure is mine, and I will try now to share my presentation. I guess it's.
We cannot see it yet.
Let's see. Okay. Can you see it now? No.
Not yet.
Now, probably.
Okay. Yes, there it is. Just wide the screen.
See, I'm doing it now.
Okay.
Okay.
Yes.
Now, can
excellent.
you see it properly? I think.
Yep.
Okay. I think this is first slide. So sorry for these inconveniences caused by technologies, and thanks, of course, Pedro and all the team of the Light Vidalia project for letting me present some research results of our group bearing on the conservation of the Canadian flora. Also, I will share some thoughts and describe, of course, ongoing efforts centered on some Canadian lotus, which is, well, probably the main subject of my presentation. Lotus, as we mostly know here in the audience, represents the largest and most widely distributed genus of the tribe Lotiae, and its main centers of species diversity are the Mediterranean and Micronesia, with about 50 species, 50 taxa in this multinational oceanic archipelago. And Lotus is also exceptional because its distribution encompasses all 5 Micronesian archipelagos plus the continental Micronesian enclave, so that I picked out some examples of Canadian lotus because, well, these in the Canary Islands encompass also some contrasting cases that illustrate, well, how challenging and how exciting is to undertake conservation efforts which are science-based in such a shifting and complex environment with so many interactions, well, going on. Since 2009, but especially in this 2017 paper, the surfing Singamian hypothesis has strived to explain the origins of the high genetic diversity detected in the Canarian and other oceanic floras as the dynamic result of admixture, introgression, hybridization going on in cycles, and also incorporating as a driving force to all those evolutionary aspects, the role of island ontogeny in the generation of the genetic diversity patterns, so that the context we're working with entails like colonizers of multiple origins in many more cases than we thought barely a decade ago. In different times, of course, sometimes back colonization of the mainland, especially from the Canaries, given the short distance to the mainland, within island migration, diversification, and recurrent and very dynamic island hopping, and accompanied, of course, by extinction, recolonization, further hybridization, and so on. So that we're confronted in conservation with a difficult landscape, because it is difficult to take good conservation action when everything is constantly taking shape, as it happens in oceanic islands, and especially in the Holocene, after a new and powerful variable has been introduced in the conservation of the forest, which is human action. Hanno and Sandrine have already referred to this powerful and very negative force in many cases. So that it's an evolution in action context where recurrent cycles of gene flow and disruptions thereof have developed symbiotically with geological ontogeny of the different islands and archipelagos, giving rise to interactions that increase or decrease genetic diversity, corresponding with cycles where this ontogeny favors gene flow or favors the development of physical or ecological barriers that impede gene flow. Furthermore, the application of molecular tools and numerical taxonomy keep revealing overlooked and cryptic species together with incipient speciation processes. And in most of these cases, these cryptic species and the overlooked ones also deserve conservation attention. And well, of course, islands keep being colonized and new endemics are generated in both older and newer islands. Evolution is not something that happened sometime in the past, but keeps developing now for us to understand it, to investigate it, and also to conserve its result. For conservation, well, there are some very basic implications. If we have to respect these evolutionary dynamics in our efforts to preserve biodiversity in oceanic islands. For instance, in situ conservation should avoid artificial mixed reinforcements in such a context of fragmentation and constant change. And of course, watch over artificial fragmentation effects, but also we should strive to maintain connectivity between the populations, connect populations, so that adaptation to changes can be fostered to generate the endemics of the future, so to speak, as it was through the past. But well, not exactly through the past, because as I said before, there's now an incipient force taking impulse since the last, well, perhaps 2,000, 3,000 years here in the Canary Islands, which is human disturbances. Because the biogeographical traits that generated this great diversity that we try to conserve in the past, like woodiness, about 72% of the endemic Canarian flora is woody. There's a high proportion of self-incompatible species, and there's of course this natural isolation and fragmentation, which is determined by the complex and changing geography of the islands. All these geographical traits and biological traits are the same ones that now constitute a threat in a context of this rapid and human-mediated change. So invasives, global changes, short time of adaptation, major habitat losses make that the network that constitutes biodiversity is breaking by the second, and probably in a few years, if we're not experiencing it already now, well, biodiversity is starting to work without rhyme or reason, losing connectivity and losing any sense. We're experiencing and witnessing, in fact, decreases of population sizes, losses of mutualisms, genetic diversity losses, reproductive problems in many of the threatened floras and even in not threatened endemics, and highest threats are starting to be very visible in all endemics, of course, especially single island endemics, the woody plants that are not adapted to this fast change rates of changes, coastal biodiversity and summit biodiversity on account of this rapid increase of global temperatures. Sorry. The genus Lotus in Micronesia reflects, I believe, to a large extent, many of the tenets of these phenomena, this surfing Singamian context. And well, in fact, the latest paper that we published, Ruth Jaén from our group, together with other collaborators, and Darío Ojeda, reflects this very dynamic context of multiple founder events, very dynamic inter and intra-island colonization, independent colonizations of the same island, followed by likely hybridization, multiple habitat shifts within a single island, and a general rejection of this progression rule because of the colonization of older islands from younger ones, and then in some cases, even mainland back colonization. within genus Lotus, this section Pedrosia includes about
We don't have your sound, Juli. We seem to lost your sound. Juli, we cannot hear you. We can see your presentation. Your sound is muted, Juli.
Can you hear me now?
Yes, we can hear you.
Okay.
But not your presentation.
Yeah. I'm going now to retake it. Where did you lose track of my presentation?
Just a few seconds in the last slide you showed.
Okay. You can see it now, can't you?
Yes, we can.
Okay. I'm trying to share the whole screen, but now you can see it?
Yes.
Okay. I was saying that section, we will restrain ourselves to the 7 endemics of the section Pedrosia that occur in the pine forest of Gran Canaria, because they have already high morphological and genetic variation. They are probably associated with independent colonizations of the island of Gran Canaria, followed by hybridization. And there's a lot of taxonomic confusion among them, as we already put forward in this old paper in 2005, that where the first author, Felicia Oliva, used especially allozymes to reveal the differences that among these 7 endemics, and to derive also some conservation implications. And well, I refer to this old technique of the allozymes because it's out of use now, but it provided a lot of good data to understand diversification in the Canaries, and these data are very often confirmed by molecular investigations based in DNA sequences or genomics. So a first question that we have for the conservation of this complex group of endemic lotus from section Pedrosia in Gran Canaria was whether a combination of morphological and molecular data could help us circumscribe taxonomic relationships. So we undertook a very wide sampling in order also to provide comparison of these 7 species from Gran Canaria with other species from El Hierro, for instance, where a Lotus SP without a clear taxonomic ID occurs. Also in La Palma, Lotus silbrandii, Lotus campylocladus from Tenerife, Lotus partioides, a strict endemic of the pine forests in Gran Canaria in the west of the island. Lotus genistoides in Gran Canaria, also in the central part. Lotus rosericius with several populations south of the island of Gran Canaria. And well, about 9 populations of lotus belonging to the section Pedrosia, which, well, we labeled Lotus SP because they have some shared characteristics between one or several of those species that I cited earlier, and we were hoping that allozymes and morphological variables could throw some light in their relationships with the rest of section Pedrosia. This is the summary of the sampling with a majority of the samples taken in Gran Canaria, which was the core of this study. This is the protocol of the morphological analysis that we ran together with the molecular analysis. And this is the basic results of this morphological plus allosemic analysis, where, well, we observe some enigmatic groupings. For instance, the Lotus SPs from El Hierro and Gran Canaria, squared in the frame under number 2 in the center of the image, are quite close together in the multivariate space. The lotuses from Tenerife and La Palma are quite close genetically, as expected, and as it is, well, confirmed recently by several works, not only in lotus but also in many other endemics. And also we obtained a very informative separation between, well, Lotus partioides plus several populations that had been termed Lotus sps, and then Lotus genistoides and Lotus orostodicius plus one of these populations termed Lotus sp. So that, well, although there were discrepancies between morphological and genetic data, and even though allozymes offered more discriminative power than morphology, the combination of both kinds of data allowed us to produce some meaningful insight. For instance, one of the conclusions of this work and its published work and of other unpublished data was that Lotus genistoides should be included within Lotus holocericius on account of the very high similarity both morphologically and in molecular terms, taking allozymes as the base data. And also Lotus campylocladus from Tenerife and Hillebrandii from La Palma are very closely related. Regarding the 8 Lotus sps from Gran Canaria, which were there in the vagaries of taxonomy and therefore were difficult to conserve, or because they had no a specific identity assigned, well, they are a heterogeneous group, because whereas 4 of them are closer to taxa, to known taxa from other islands in the Canarian archipelagos, 3 of them group with Lotus partioides, another one with Lotus orostodicius. There are still some uncertainties regarding these ones that impede their inclusion in conservation strategies. It is good to see that a work published in 2012 by Isidro Ojeda and collaborators greatly confirms many of these insights. This paper took 4 nuclear regions and 3 plastic regions, and in fact confirmed the hypothesis of the multiple origins of the lotus from the pine forest in Gran Canaria that we produced in this allozyme work. It confirmed also the context of highly dynamic within island migration in Gran Canaria, and very likely confirmed hybridization following this context of within island migration between species whose origin is in different islands of the archipelago sometimes. Okay, and another case, another entirely different case within section Pedrosia is that concerns Lotus kunkelii, which is a coastal endemic with just a single population in the Hinamar site of scientific interest in the east of Gran Canaria Island. It is absolutely critically threatened, and well, I pick out this example because I think it illustrates one of the points of interest of this workshop of the Life Vidalia project, which is how different lines of evidence can conform a holistic conservation plan for a highly threatened endemic with just one known population in one island. So a first question regarding these Lotus kunkelii regarded taxonomy, because in the early 2000s there still was the doubt of whether Lotus kunkelii was a subspecies or could be a subspecies of Lotus lancerotensis. And this posed a very important conservation challenge, because well, given the high amount of endemic species here in the Canaries, subspecies do not deserve official protection conditions. So if Lotus kunkelii was a subspecies of Lotus lancerotensis, probably the Canadian government wouldn't have provided conservation measures for these very, very critically threatened species. And again, allozymes showed quite distinctly that Lotus kunkelii was quite far from Lotus lancerotensis genetically, and much closer to another endemic from Gran Canaria called Lotus arinagensis, whose populations lie not far from the single population of Lotus kunkelii. Based on this work, published in 2006 in the American Journal of Botany, the Canadian government proposed and published a recovery plan decree where they recognized the results of this molecular study there in the area squared in yellow, and they admit that these results give rise to the declaration of this species as critically endangered, and to propose this recovery plan. And luckily, well, further analysis with a more comprehensive sampling of the Lotus in Macaronesia have confirmed the evidence provided by allozymes. Lucky to us. So Lotus kunkelii is indeed a good species, also basing ourselves on several sequence regions, and also in this latest work with the genus published very recently in Molecular Phylogenetics and Evolution, we confirm that Lotus kunkelii is very, very far from Lotus lancerotensis phylogenetically, but it is closest to the Tenerifan Lotus mascaensis rather than to Lotus arinagensis, which was our hypothesis in the allozyme work. And also these works, on account of the dating of the ages of the species, reveal that Lotus kunkelii is not a relic species, but indeed a very young one, corresponding to the late Pleistocene, probably just 0.2 million years ago. So probably it will be of interest to the participants in the live VIDALIA project to know the objectives of this recovery plan and how they are being accomplished by the Canadian government, and with a very protagonist role of our institution, because we, as many of you know, belong to the Cabildo de Gran Canaria, which is the local administration in the island of Gran Canaria, and we have official competences regarding the conservation ex situ and in situ of the endemic diversity of these islands. So there are two objectives in this recovery plan. The first one is this one you can see in boldface, to increase the current number of individuals of the species in order to establish a population of at least 5,000 reproductive individuals distributed in 3 nuclei, as well as to have the instruments for its ex situ conservation. So the action 1 and 2 within this objective 1 relate to seed collection and the collection also of vegetative samples in this only known population of Lotus kunkelii, and to send at least 2,000 seeds to other seed banks for its short and mid-term conservation. In fact, this is a task that we're undertaking since very many years ago, even before Lotus kunkelii was declared critically endangered, and even before it had a recovery plan. You can see a snapshot corresponding to the 26th of May of 2016, where several seeds of Lotus kunkelii were collected, and a more recent one to your right, taken in April 9th, 2021, with the project Netzgendem, which among other lines of action, it encompasses the collection and the preservation of seeds of very many endangered species. And regarding to Lotus kunkelii, this counter of the seed counter indicates you that in the last expedition, 3,148 seeds were collected from that single population, which is extremely good news, at least for the ex situ preservation of the species. Still within objective 1, the action 3 corresponds to creating seedling stocks that could allow us to obtain seeds when the reproductive turnover of the natural population is on the wane, and also to carry out the needed actions to increase the germination and rooting rates of the vegetative material, as well as the acclimation of the plants in cultivation before their reintroduction. I was glad to hear Sandrine when she said that very often reintroductions corresponding to seedlings work much better than seeds. So we're in the good way, as I will show later in this presentation. And also action 5 refers to cultivating the seeds and vegetative material collected to obtain living stocks to be used in the reinforcements and reintroductions. And I'm proud to say that many of these actions are being undertaken by our institution in connection to the greenhouses of the Cabildo de Gran Canaria, which is the local administration we belong to, where they have the mother plants from the natural population of lotus, and a whole collection of individuals reproduced from those mother plants taken from the natural population. So probably prospects of genetic contamination are not even probable in this case. The action 6 in this objective 1 was to carry out reinforcements and reintroductions in this only known population at Ginamar, and action 7 to 11 are related to monitoring, informing, and identifying the plants, together with also training the rangers to differentiate Lotus kunkelii from Lotus glaucus, which occurs quite near the wild population of the endangered endemic, and well, that can beget a risk, because, well, if Lotus glaucus is confused and they take seeds to the greenhouses, you can imagine the drama that we would have. So this is just a very simple scheme of the reinforcements and reintroductions that have been developed from 2009 till now. Some of them have had greater success, some of them lesser success. There are neighborhoods that just have one specimen after some years of growth in that wild environment, but other neighborhoods have very big specimens, and they are reproducing without any problem, as accounted by all these reports that you can, if you're interested, you can download them or see in the website of the Botanic Garden Viera y Clavijo, where I work. So
Juli, sorry
yes.
to interrupt, Juli.
Yeah.
I don't know if your slides have stopped or if you.
No. No. Can you see them changing now?
No. We are still on the in the case of Lotus kunkelii, a holistic conservation plan.
Oh, okay.
So
no. Then
sorry, I thought you were using the same slide.
No. I've been showing a lot of slides. Let me reload again. I think Sandrine had some similar problem, but
she did. Yes.
Okay. Let me see if.
Yeah.
Now I'm opening the. Okay. Okay. Can you see it? Can you see it now?
We can. Yes. Okay.
Okay. Now I will put it
be
in.
Aware.
I'm
be
already
just aware of your time, please.
Yeah. I'm already finishing, but I didn't know you were not seeing the. Can you see it now?
Yes.
Can you see it changing?
Yes. Yes.
Okay, so this is the counter showing the 3,148 seeds that we collected in the last expedition, together with a snapshot I commented from 2016. The greenhouses in the Tafira area that belong to the Cabildo and work in coordination with our botanic garden to develop all the actions within this objective one. And can you see them now? The reintroduced
yes.
plot, the green dots correspond to the 15 reintroductions, 15 neighborhoods, which are reintroduced together with the two original wild distribution patches, just two patches, one smaller than the other. And as I said, in some cases, this just one specimen in the 12th neighborhood, there has been post success in the rooting of the plants, but in other cases, like to the right in the 13th neighborhood, there are big specimens which are producing good seed. And just to finish with something funny, well, objective 2 of this recovery plan is aimed at promoting the accomplishment of the regime of uses of the Hinamar scientific interest site, and also to undertake a dissemination program about Lotus kunkelii. And this is one small contribution from the garden to this dissemination program, which is the song dedicated to Lotus kunkelii within this project of the musical herbarium that we're taking with the El Cancionero Isleño. So I'm leaving you with this song dedicated to Lotus kunkelii as part of this dissemination program. It will be barely 2 minutes, and after that I can take your questions. Can you see the screen?
Yes, we can.
Okay, then can you listen to it?
Yes.
Okay, it's in Spanish, but anyway, it's a nice way to convey this is the original population, the landscape where the lotus is. This is shot in the original population of lotus in the natural environment.
We cannot see the video.
Oh,
just the music.
Now, can you see it now?
Just we can only listen to the music.
Well, anyway, if you're interested in seeing the video, you can download it or visualize it from the hyperlink I included in the presentation, or visiting our YouTube page in the garden where videos for 17 other species are available. So
excellent.
Anyway, that was my contribution, and I will take questions now or complaints.
Okay, thank you very much, Juli. Yes, we can. Maybe it's probably a good idea to have your presentations and then send it back to the participants in our workshop so they can take a more closer insight on your presentations. Thank you very much, Juli. It's been very interesting to understand how all this molecular data influence the conservation approach to the species, and well, we've been a bit beyond our time with the technical issues. Anyway, we will. I'll just put you one question. What is, in your opinion, what is the biggest challenge you have encountered in the conservation of this species of lotus, and what are your thoughts on this genus long-term conservation?
Well, there are various issues, of course. Can you listen to me?
Yes.
Okay. Okay. There are various issues. For instance, as regards this complex of species, which sometimes contain taxonomically conflictive species. One of the issues is the very dynamism of evolution in oceanic islands, because we have to preserve species, but I think the most important thing is to preserve the processes that will beget endemics in the.
We lost your sound, Uli. Your sound is muted. Hello, Uli.
Can you listen to me?
Yes, I can. I cannot see you, but I can listen to you.
Yeah. I cannot connect my camera. I don't know why.
Okay.
But anyway, I was saying that in the cases of these species which have a single population or occur in a single island, there's also a very nice prospect that molecular data can offer, which is dating these endemics. As I said in my presentation, Lotus kunkelii is a very young endemic, so it is endangered because it's a baby. So probably what we have to do with these very young endemics that occur all through the archipelago is to protect them ex situ, but also to reintroduce them in areas near their original wild occurrence area in order to foster connectivity. And probably the same happens with other young endemics that need to have the opportunity to expand. With older endemics or with relict endemics, probably the situation is very different because they normally have reproductive problems that may advise, well, it's much better to opt for an ex situ conservation strategy or for major weight of ex situ strategies, and then to protect the habitat of these species. But overall, I think in order to protect the capacity of the lineages to evolve, it is very important to maintain the connectivity between the populations so that they can establish or discontinue gene flow when the evolution of the habitats allows it or disallows it.
Okay, Juli, thank you so much for your presentation and participation in our workshop. It gave us a really another interesting view over this problematic of species conservation. Thank you, Juli. Thank you very much.
Thanks to you, and sorry for the technical inconveniences.
It happens. We all know it happens. It's okay. Okay, so we will move forward to the next presentation. So I will now present Francisco Fernandez. He's a senior technician in the Institute of Forests and Nature Conservation in Madeira, Portugal. As a technician, he has worked in live projects dealing with plant conservation through inventory, monitoring, propagation, and population reinforcement. He was involved in the establishment of a seed bank for indigenous plants and a laboratory for in vitro culture of endangered species at Madeira Botanical Garden. So he will now present his presentation on how to improving the resilience of Porto Santo Island's dune ecosystem to the impacts of climate change, this under Project Life Dunas. So, Francisco, are you there?
Yes, I'm there. I'm here. So I suppose you are viewing my presentation. Is okay?
We are viewing your presentation and also the comments you have on the slides. So maybe you can use the so
I'm trying to move out the comments because it's only for me, of course. I'm trying to. A moment, please.
Yes, take your time.
My friend is helping me my colleague to try to resolve this problem. Is
okay. I don't have your, maybe your camera is off.
Yes.
I cannot see your presentation right now.
And now you.
Oh, it's coming. Yes, I think.
With the comments or not?
I am still. I cannot see your presentation. You have your camera on, do you?
Yes, I'm trying to. Yes, my camera is off now. My camera
off.
Only the sound. Yes.
And you shared your screen?
Yes.
Okay. Yeah.
It's okay. No.
It still has the comments.
Still has the comments. Okay.
But it's okay.
It's okay.
It's okay. Thank you very much for accepting our invitation, Francisco. Go ahead.
Okay. In this presentation, I will highlight several actions in the context of life dunas that aim to consolidate with native vegetation a rebuilt dune on Porto Santo Island. Porto Santo Island belongs to a Madeira archipelago that was officially discovered by Portuguese Gonçalves Arco and Tristão Vaz Teixeira around 1418 during the period of Portuguese discoveries and exploration of West African coast. Historical knowledge based on old texts make global picture of original flora of Porto Santo with few tree species. First reports from Porto Santo Island flora by Luís Cadamosto in 1445 mentions presence of the Dracaena draco. This picture in the slide is not Porto Santo but Madeira Island. Leda Frutuoso in the 16th century reports a very peculiar vegetation, namely dragon trees and junipers that we currently think to correspond to Dracaena draco and Junipers turbinata canariensis, as well as Barbusana, Plonus barbujana, now extinct in this island. At present, dragon tree species no longer exist in natural conditions in Porto Santo, and this only preserved in the coasts of farmers of the city, and only 3 juniperus plant remains in cliffs of Pico Branco. The introduction of rabbits in the past was a major threat to indigenous flora and vegetation. It seems that Bartolomeu Pastrello, the first Capitão Donatório of Porto Santo, released a female rabbit and her offspring, but within a short time, these animals multiplied in such a way that he could not sow or plant anything that was not immediately devoured or spoiled by them. Nowadays, a rabbit is still a problem for native flora and vegetation on this island. Due to the lack of vegetation as a result of human activities, since the settlement, several species were introduced for use in the agriculture and for fuel. In this way, a rhododendron was introduced in 1783 to delimit agricultural land. Tamarix gallica by 1844 for fuel. Elaeagnus angustifolia and Carpobrotziduli perhaps for use in gardening.
Francisco, sorry to interrupt.
Yes.
It seems like some of the participants are not seeing the slides. I can see them, but it seems like not everyone is able to see. But some have already are seeing it again. So I would say that please go on. Please go on.
I don't know what happened. I can give the after the slides. No, I don't know.
Send the presentation. Okay. Yeah. Well,
okay.
Please go on. Sorry for interruption.
Okay. Thank you. Only dunes on the beach. Sand. Rhododendrons and Carpobrotzidulus present serious threats to natural diversity of this very sensitive area. So this is a slide where you can see the dunes with Rhododendrons, Carpobrotzidulus, as well as Calistesia soldanella. Climate change scenarios analyzed and discussed in Madeire's Regional Strategy Adaptation to Climate Change, approved in 2015, foresee for Porto Santo the increased frequency of extreme weather events. To prepare Porto Santo coastal ecosystem to climate change, the Secretaria Regional do Ambiente, Recursos Naturais e Alterações Climáticas is implementing a live project named Life Dunas. Life Dunas is a project developed under the Life Climate Change Adaptation sub-program whose projects aim to increasing resilience to climate change. This project targets improving resilience of a dune ecosystem to climate change impacts. These impacts require adequate measures to avoid further degradation of the coastline and prevent further risks and damage on the local environment and socio-economy. Project area is close. This project area of Life Dunas is close to Calheta in the extreme west of Porto Santo Island. Porto Santo Island is there, is in the west part of the Porto Santo. Main operational objective of Life Dunas: restoration of a dune that was depleted in the 1950s. This action includes two phases. The first includes geomorphological works using about 19 90,000 cubic meters of sand degraded from near shore and sandbanks, without compromising regular coastal dynamics. The second aims coastal restoration based on plants of native flora and control of invasive alien species on the wider buffer area. Reuse of upper dune for agriculture with production methods and practices to improve dune resilience by reducing losses of wind erosion and devolution of sand to the dunes. Improving awareness raising in the local population and users of sand dunes. The blue circles indicate the area where the project is being implemented. This is a view of Porto Santo, and the circle indicates the area of the project. In this area, there is a big depression caused by sand extraction for construction of Porto Santo Airport in the 1950s. So we can see the disruptive primary dune that corresponds to intervention area is this area here. This is the primary dune there, and this is the project will the rebuild of the dune in this area. The project includes several preparatory actions important for correct implementation of the conservation measures. These include updating natural and invasive flora as well as vegetation cover before implementing the project actions on the ground. This knowledge was important to prepare operational plans to control invasive alien species and habitat restoration in the buffer area and rebuilding dune. This knowledge acquired before project implemented is also important to evaluate actions. So this area is for the rebuild of the dune, and this is the buffer area that was implemented actions to control invasive species of flora, as well as to recover native flora. In this event preparatory actions, flora from all area was inventoried. 23% of the flora of this area is non-native, and we only found 2 endemic species of Porto Santo, Lotus glaucus floridus and Lotus lolianus. Most of the flora covered species are native. We have, from the total of 44, we have 24 species native, 10 exotic, and only 2 endemics of Porto Santo. Methodology of the inventory. The buffer areas, these areas, and the other correspond to lateral to the depression, to this depression area, that the dune will be rebuilt, were divided into 10 by 10 meters squares matrix, as we can see in the picture, in this picture. Within this matrix, alternating squares were used to carry out inventories. In the second picture, we can see how we make inventories. In this, we can this picture that is illustrating how we can make the inventories in the field. To which species was applied the Brown-Planck Abundance Index. This is an example of the output that we aim to for all species, native and exotic, of the project. Dark green squares present greater cover of Lotus glaucus floridos. This data will be used to assess the evolution of the flora and vegetation over the project. This phytosociological data was the bibliographic data used in the selection of species to cover the dune and also the restoration of the surrounding areas, buffer areas. The rebuilt dune corresponds to a primary dune dominated by Euphorbia paralias, Polígono marítimo, and Calistegia and Soldanella. So all the restoration habitats will be based in the bibliographic data with phytosociological studies. Next, I will describe implementation actions of life dunes. To promote dune cover, seeds from native species of flora and identified as being part of the local habitats were collected. Propagation will occur in the local nursery. Sand and peat will be used in order to avoid the introduction of invasive species in the plantation area. So you can see here a nursery in Porto Santo, where the indigenous plant will be propagated, and this is Lotus glaucus floridus in the nursery. Control of invasive alien species. Buffer areas was divided for testing different ways to control rhododendrons. The control of rhododendrons in the area D1 will be done through successive cuts with the use of brush cutter next to the rhizome. Cuts must be made whenever necessary so that the shoots do not exceed an average height of 0.3 meters. This successive mechanical cut aims to deplete the rhizome reserve food so that the plant does not sprout. Remaining area of D1 will be used with the rhododendron control tests, namely shading by geosynthetic screen, sodium chloride after cutting, linseed oil after cutting. The area D2 will be used with the rhododendron control tests, namely shading by Serapilera, that must be completely covered with sand with a height at least 0.1 meters. This control test aims to find the best practice control method that will be applied on the rhododendrons in this project. For Capabrotedoulis, we will manually remove all plants and place them on plastics to be dried, leather being removed from the intervention area. Concerning habitat restoration, species used in the recovery of the primary dune and restoration of the secondary dune will be based on phytosociological data, as I said in the previous slide. The species Lotus lowlianus and Lotus glaucus floridus are present concerning in the secondary and in interdunar areas. It seems that the protection of these endemic species in this habitat depends on the preservation of this specific habitat, since they appear on the back of primary dune. So this is the dune that will be rebuilt, and the plants that will be consolidated the dune. Calistegia, Polígono marítimo, and Euphorbia paralias. We use only these species to consolidate the dune that will be rebuilt in this project. These lots are this is examples of Lotus glaucus floridus, this one, and this is the Lotus lowlianus, all both endemic to Porto Santo. For the restoration of vegetation, planting will be carried out in species areas, circles 1 meter, separated by 2 meters. That all species will be planted in the rebuilt area in circles of 1 meter, and separated by 2 meters. Species compositions are different and depend of the area where there will be planted. So we use in checks in these circles in primary dune we use a mix of Inflorescens, Polígono Marítimo e Calistenia Soldanella. And secondary dune and the interdunal space we use a mixed too. It's different mix in the different parts of the areas. All plants will be planted in palisades, at least those sided to predominant winds on the island of Porto Santo, to be protected by from the wind. That is sometimes is very strong in this island. So, thank you for your attention, and I'm waiting for any question. Thank you.
Thank you, Francisco, for your interesting presentation, and congratulations on your interesting project on renaturalizing that sand system in Porto Santo. So
I'm trying not to put the camera.
Okay.
Oh, okay. No. Yes.
That's right. There you are.
Thank you.
Thank you. So, so what do you think are the greatest challenges in conserving these coastal systems of dunes? Is it logistics, or maybe the public acceptance of the inhabitants? What do you consider to be the greatest challenges?
I think are very challenges. First is the climate change. I think the waves, big waves, wind. I think it's a problem. We in some years ago there was storms that we can see the problem with in the near future with climate change with the dunes. Another is the persons, because in Porto Santo Island in the summertime the population is 3 times more than the original population. So the impact of is very important, and I think this is the most important impacts in this type of ecosystems.
What measures are you implementing towards the people? So I imagine you don't like them to go around in the dune area.
It's not our area of the project. Our area is only vegetation, but I know we put I don't know the corridor. I don't know in English. So os corredores. No, and I don't translate. I don't know to translate.
The corridors, so where people can go by the dunes.
Yes.
Okay.
Yes.
Okay. Interesting.
And of course, wearing in value education.
The environmental education. Sure.
Yes.
Okay, great. That's good. So we are on our schedule. So it's 12 o'clock. It's time for our lunch break. We'll be back at 14:20 to go on with our works and to present the works of Life Vidalia. and to do this virtual visit on some of our areas and works. We have prepared a few videos for you. So thank you very much, Francisco, for your presentation, and also to all the other speakers and all the people that came to watch our workshop. Thank you very much.
Thank you for inviting me also, and see you.
It was a pleasure. Thank you. So see you later at 2:20. Bye bye. Thank you very much.